Hello, I'm Joanne Lupton, Professor of Nutrition at Texas A&M University and Team Leader for Nutrition and Physical Fitness at the National Space Biomedical Research Institute. Long-duration spaceflight poses a number of important challenges to human physiological systems. This presentation focuses on how nutrition and exercise can be used as countermeasures to some of the more critical risks and emphasizes the importance of optimal nutrition and physical fitness in health, both in space and on Earth. There'll be 4 parts to this presentation. First, the importance of nutrition to manned spaceflight. Then, 2 key areas for nutritional countermeasures, and those include muscle wasting and radiation-enhanced cancer. And it's important to consider the whole person, since when we eat, the nutrients that come into our body are not targeted just to one specific organ. And finally, I'll close with some applications to Earth. What are the problems encountered in space that may be positively affected by proper nutrition and exercise? Well, all of them, from radiation-enhanced cancer to psychosocial factors. But we'll concentrate on 2 in particular, muscle atrophy and radiation-enhanced cancer, and talk briefly about the problem of bone loss. How are risks identified and prioritized? Well, they are defined on the Bioastronautics Roadmap. This is an iterative process. It's a joint effort of NASA and NSBRI. And using this process, 45 risks have been identified. Now, each of these risks is given a priority score from 1 to 3, depending on the particular mission, whether it's the International Space Station, a trip to the Moon, or an exploration mission to Mars. And on a Mars mission, which is what I'm showing here, nutrition and radiation-enhanced carcinogenesis are both a #1 priority. Reduced muscle mass and strength and increased bone loss are a second priority. But still, just being one of these 45 risks makes them very important to spaceflight, very important to NASA and NSBRI. The first issue that I'd like to talk about is muscle wasting. We'll be discussing 2 types of countermeasures, 1 to muscle wasting and 1 to radiation-enhanced cancer. The issue that comes with muscle wasting is shown on this slide. The problem is that protein synthesis for muscle must equal protein breakdown, and it doesn't during long-duration spaceflight. It doesn't because there's an increase in muscle loss, and that is due primarily to inactivity, to less than optimal nutrition, and to stress factors like increased cortisol secretion. In order to balance that, we need some countermeasures. We need to optimize nutrition, we need to have a good exercise protocol, and perhaps even use anabolic agents. We have 2 key projects on our team that address muscle wasting. The cornerstone project is the Wolfe bed rest study, and this study tests an essential amino acid supplement. We have a second study that uses the same protocol intentionally as the Wolfe bed rest study so that we can compare one study to another, but this study adds exercise and timing. This slide shows the protocol for the Wolfe bed rest study, which is a 28-day bed rest study, bed rest being a good analog for spaceflight. On days 1 through 5, there is a diet stabilization period where lean leg mass is measured by DEXA, calf volume by MRI, and there are strength tests that are performed. On day 6 is the first tracer study. The tracer study looks at protein synthesis. Days 7 through 33 are strict bed rest, and a number of experiments are done during that time. Then on day 34 is the second tracer study to see the impact of the bed rest period on protein synthesis. Days 35 through 38 are reambulation, and again we look back at the effect on lean leg mass and calf volume. Now, there are two groups for this bed rest study. One of them is bed rest alone with a placebo, and the second group is bed rest plus amino acids, and that's the intervention. This shows some data from that experiment on leg muscle mass change after 28 days of bed rest. The yellow bar shows the results from the essential amino acid supplement, and the white bar shows the placebo. So we can see here that the essential amino acid supplement helped to maintain leg muscle mass, whereas the placebo group lost muscle mass during the 28-day bed rest study. In contrast to that, this slide shows the loss of leg strength after 28 days of bed rest. Here, the essential amino acid supplement was not able to maintain the leg strength during the bed rest study. However, it did succeed better than did the placebo group. So to summarize the Wolfe bed rest study then, an essential amino acid supplement maintained muscle mass during 28 days of bed rest. An essential amino acid supplement ameliorated but did not maintain muscle strength. We then moved into a second study after this Wolfe bed rest study, seeing that an essential amino acid supplement alone would not maintain muscle strength. We used the same protocol, but this time added an exercise component and a timing component. And what I mean by timing is that there was an optimal time between ingestion of the supplement and performance of the exercise. This study was done by Dr. Carmen Castaneda-Shepa at Tufts. The purpose of the Tufts bed rest study was to determine, is there a synergistic effect on muscle atrophy of an amino acid supplement combined with resistance exercise and also combined with the appropriate timing of the supplement intake to the exercise. There were three groups for the study. One was an amino acid only group. The second group combined the amino acids with resistance training. And the third group had the amino acids, the resistance training, and the optimal timing between the two. It's important to point out that all subjects received only 85% of their energy requirement. And this is designed to mimic what actually happens in space, where astronauts do not eat all of the energy requirement that they should. The exercise component was a 1-hour session, 6 days a week. It worked all of the major muscle groups, and it used both upper and lower body. So one day would be the upper body, the next day would be the lower body. The exercise intensity scale was used to come to a completion of active, moderate, or high intensity exercise. Study outcomes included body composition, resting energy expenditure, mid-thigh composition, and muscle strength and function. This slide shows the reduction in thigh muscle area after 28 days of bed rest. The green bar is the amino acid only group. The yellow bar is the amino acid plus the resistance training. And the orange bar is the amino acid, resistance training, and optimal timing. And as we can see here, the muscle area was reduced about 12% even with the amino acid supplement. Resistance training, however, mitigated the reduction in muscle area. Now we're looking at reduction in lower body strength. Lower body strength was reduced by 24% in the amino acid supplement only group, shown on the green bar. But resistance training mitigated this reduction, shown in the yellow bar, and on the orange bar was the best combination. That was timing of the ingestion of the supplement to the training, and that further enhanced the protective effect. So what we see here having to do with muscle wasting then, and summarizing the two studies that I've just shown you, an essential amino acid supplement has a beneficial effect. Resistance exercise has a beneficial effect, and the two combined are better than either one on its own. And the timing of the supplement intake to exercise is also important. These help to protect against loss of both muscle mass and muscle strength. The next countermeasure that I'd like to discuss is radiation-enhanced cancer. As I mentioned previously, risks to personnel in space from naturally occurring radiations are one of the most serious limitations to human space missions. And the major risk from radiation exposure is later cancer development. This has a priority #1 rating on the bioastronautics roadmap. It will become even more important when we return humans to the moon and in preparation for a human exploration of Mars and other destinations. Of all the cancers, colon cancer incidence is a likely target for radiation enhancement. Colon cancer is the second leading cause of cancer deaths in the United States today, and it strikes men and women pretty much equally. Colon cancer is a multi-step process from a normal epithelium all the way through to metastasis. So, for example, all the epithelial cells in the colon are only one cell layer deep. They are arranged in these patterns shown here called crypts. And at the very base of the crypt are the stem cells that produce daughter cells. Those daughter cells then can migrate up the side of the crypt, differentiate, and towards the top of the crypt, they will undergo a process called apoptosis or programmed cell death and get exfoliated into the fecal stream. We are able to recapture those cells that have been exfoliated in fecal material and measure messenger RNA on those cells. Thus, we can do this at every stage of the tumorigenic process, from the very normal epithelium to dysplastic crypts, up to what are called aberrant crypts, which is partway through the process, to adenomas, and all the way through to carcinomas. And by using this process, we can see changes in colon cell expression patterns over time. We had two hypotheses for our NSBRI countermeasure studies. The first was that a diet containing fish oil and pectin will be protective against tumor development compared to corn oil and cellulose. This hypothesis was because we had previously shown that using a chemical carcinogen, this diet was very protective. And we hypothesized that it would be protective at every time point during the tumorigenic process, at initiation, at the promotion stage, and at final tumor development. We further hypothesized that a non-invasive technology, which I've just explained to you, can be used to track changes in gene expression from colon cells over time. To test these two hypotheses, we used 560 Sprague Dawley rats in a 2 by 2 by 2 by 3 factorial design. That is, they were receiving either corn oil or fish oil, either pectin or cellulose. They were either irradiated or not with 1 gray iron ions at Brookhaven National Laboratory, and they were terminated at 3 time points: the initiation stage, the promotion stage, and the final tumor stage. During this experiment, fecal material was collected over time from a subset of the rats, and it was analyzed for changes in gene expression profiles using microarray technology. This slide shows the very earliest stage of the tumorigenic process, the initiation stage. This is when DNA damage occurs. Oxidative damage is measured by an adduct called 8-hydroxydeoxyguanosine. This is the type of adduct that would occur, the DNA damage, in the presence of radiation. What we're showing here on the green bars is that the fish oil diet, rats receiving that particular diet, had a lower level of these damaging, oxidative DNA damaging agents than did the corn oil group. At the middle stage, when these aberrant crypts appear, again, the fish oil diet had a lower level of these aberrant crypts, which can become colon tumors both in rats and in humans, than did the rats receiving the corn oil treatment. And finally, at the very last stage, the colon tumor stage, the fish oil fed rats had a lower proportion of tumors than did those fed corn oil. We also managed to absolutely track these changes in gene expression profiles using our microarray profiles. For example, we could see the effect of diet, the effect of irradiation, and the effect of time in terms of differentially expressed genes. In other words, some genes were overexpressed or underexpressed. 387 of them as a function of diet, 125 as a function of irradiation, and 369 as a function of time that we were able to distinguish. What this slide shows, and I think it makes a very important point, is that if we pick just one of the genes that was upregulated significantly during this process, in this particular case, I'm picking interleukin-1 beta, because this particular protein supports or promotes colon cancer development. And what we show here is that if we were to ask the question, does fish oil or does corn oil upregulate the expression of this particular gene, the answer to that question would be, it depends. It depends on the time that we're looking at. So if you look at the earliest time point, around 7 weeks, you would say that the fish oil, shown in the green line, would upregulate the expression of this gene. But if you looked at around 13 weeks, there's no difference between the expression from those two diets. But at the last point, at the tumor endpoint, the corn oil cellulose diet greatly upregulated the expression of this gene. And the point that I'd like to make on this particular slide is that one snapshot on gene expression profiles does not provide accurate information. One needs to look in the manner that we did at gene expression profiles over time. So to conclude then about the radiation-enhanced cancer, and to summarize from this, a diet high in fish oil and pectin can protect against radiation-enhanced colon cancer, and a non-invasive technique can be used to monitor colon gene expression profiles over time. Now I'd like to turn to the second to last point that I'm making, and that has to do with considering the whole person. Here the issue of considering the whole person is this. If you were to have, for an example, the amino acid supplement that I mentioned previously from the 2 bed rest studies, it is possible, potentially possible, that this amino acid supplement taken to protect against muscle wasting could actually exacerbate bone loss. In some instances that have been documented, a high protein diet, perhaps through an effect on metabolic pH, causes leaching of calcium from the bone. So this is important to follow up. In contrast to that, feeding the fish oil supplement, as we did for the radiation-enhanced colon cancer studies, is more likely to protect against muscle loss. This could be through an effect on decreasing protein breakdown rather than just increasing protein synthesis, as we've seen. Nutrients are obtained from food, and inadequate food intake results in inadequate nutrition. So in the Tufts bed rest study, where energy intake was intentionally 15% lower than required, the amino acid supplement was pretty much ineffective. It's very important for astronauts to be able to obtain all of their energy requirement in space, so that amino acids, for example, are not used for energy. Instead, they're used for protein synthesis. Not only are these countermeasures important for space travel, they are also important on Earth. For example, sarcopenia, which is muscle wasting, is very high in many bedridden patients and in the elderly. These countermeasures, such as amino acid supplements, resistance training, and the timing between the two, can be very helpful in helping to decrease the incidence of sarcopenia. Similarly, with countermeasures against radiation-enhanced cancer, a number of individuals have an increased risk for cancer development due to radiation, including radiation workers, those who are undergoing radiation treatment for cancers, individuals predisposed to colon cancer. The diet that works on Earth also works in space, and vice versa. And the non-invasive techniques that we've shown with microarray can detect changes in gene expression before an actual tumor occurs. So this can be used for early detection. So to conclude, nutrition and physical fitness impact every physiological system in the body. The overall goal then of the nutrition, physical fitness, and rehabilitation team is to optimize these systems through both diet and through exercise.